This is Heliox, where evidence meets empathy. Independent, moderated, timely, deep, gentle, clinical, global, and community conversations about things that matter. Breathe easy. We go deep and lightly surface the big ideas.
Welcome back to the Deep Dive, where you bring the sources and we unpack the core insights. You sent us a really fascinating scientific paper today, digging into one of the biggest puzzles of the pandemic. Why does COVID-19 hit some people so much harder than others? We're diving deep into the world of neutrophils, those speedy first responders of your immune system, and exploring the surprising possibility that SARS-CoV-2 might actually be manipulating them directly in severe cases.
That's right. The source we're looking at, a study by Shia and colleagues, it zeroes in on the unique immune signature of severe COVID-19. It focuses particularly on neutrophils and a specific subset known as myeloid-derived suppressor cells, or MDSCs.
Right, MDSCs. So our mission today is to break down exactly what they found. What's different about neutrophils in severe disease? Could they actually predict who gets sicker? And I think the most compelling question this paper tackles, does the virus itself directly hijack these cells?
Exactly. By the end of this deep dive, you'll have a clear picture of what this paper discovered about these immune cells and their potential role in the worst outcomes of COVID-19.
Okay, let's unpack this then. We know that severe COVID-19 isn't just about the virus replicating. It often involves an immune system response that seems to, go awry. What did this paper confirm or maybe add about the overall immune picture in severe cases?
Well, the study first reinforced a pattern that was already being seen. Severe COVID-19 is marked by unusually high numbers of neutrophils, that's called neutrophilia, and low numbers of lymphocytes, known as lymphopenia.
Okay, high neutrophils, low lymphocytes.
Yeah, and this kind of imbalance isn't unique to COVID. You see it in other severe infections, too. The source notes that just having neutrophilia, that alone was already considered a predictor of a poor outcome.
Okay. But the paper doesn't stop at just the total count, does it? It really focuses on a specific type of neutrophil.
Precisely. They turn their attention to polymorphonuclear myeloid-derived suppressor cells, PMN MDSCs. Bit of a mouthful. Now, think of these as a sort of specialized group within the broader neutrophil family. They're known for one key job, basically putting the brakes on T-cell responses.
And those are crucial for fighting viruses.
Exactly. The paper highlights LOX1 as a key surface marker that helps identify these specific suppressive human PMN MDSCs. These cells, they show up in various diseases where the immune system is involved, not just infections.
And when they looked at actual patient samples, these were from hospitalized patients back in 2020, right, before vaccines were widespread.
That's right, pre-vaccine era. They found that these suppressive LOX1 marked neutrophils, along with a group called low-density neutrophils, or LDN.
LDNs, okay.
They were definitely more abundant in patients with COVID-19 compared to healthy people. But here's the crucial finding. The numbers of total neutrophils circulating LDNs and these specific PMN MDSCs were significantly higher.
Much higher.
Yeah, significantly and much higher in patients with severe COVID-19 compared to those with mild or moderate disease right at the time their samples were taken. They also saw that levels of something called elastase, which is a protein released by neutrophils,
Elastase.
that correlated with the levels of these cells. This suggests increased degranulation, basically. Neutrophils releasing their internal contents was happening more in severe cases.
Okay, so more spilling their guts, basically.
Sort of, yeah. And confirming the overall picture, T-cell frequencies were indeed lower in the severe patients, contributing to that lymphopenia we mentioned earlier. They also noted that neutrophils expressing both LOX1 and another marker called PDL1 were particularly abundant in severe cases.
PDL1 too.
Yeah.
Okay, so we see these specific neutrophil differences during severe disease, but here's where it gets really interesting and potentially useful.
Right.
Could we spot these specific problematic neutrophils early on, maybe before someone develops severe symptoms, as an early warning sign?
That was a key question the researchers wanted to answer, a really important one. They looked at samples taken very early in hospitalization, like within the first 3 days. And then they tracked how severe each patient's disease subsequently became.
Okay, so looking forward.
Exactly. And what they found was quite striking. In the patients who later went on to develop severe COVID-19, their absolute neutrophil count, the ANC.
ANC, got it.
It was already significantly higher during those very first 3 days compared to patients who stayed mild or moderate.
Wow.
So this suggests this high neutrophil count isn't just a consequence of severe disease. It might be present right at the outset, predicting trouble.
Okay, so just a high count early on is a signal. That's interesting. What other early markers did they identify that were linked to later severity?
Well, they found higher levels of reactive oxygen species, ROS generation by neutrophils early on.
ROS, those are the damaging molecules, right?
Yeah, think of them as sort of chemical sparks neutrophils can produce, which can cause damage. And significantly, they also saw a much higher expression of neutrophil PD-L1 during those first 3 days in patients who later got sicker.
And PD-L1, just to quickly define it, that's like a molecular stop signal for T cells, right?
Exactly. PD-L1 is a molecule on the cell surface that binds to PD-1 on T cells, essentially putting the brakes on the T cell's activity. It's part of a pathway the body uses to prevent the immune system from overreacting, but, pathogens can sometimes hijack it.
I see.
And the study found that neutrophils expressing both LOX1 and PD-L1 were particularly abundant in severe COVID-19 cases. This suggests these double positive cells might be key players in the immune suppression.
Okay, so LOX1 and PD-L1 together.
Yeah. Then they used a predictive model, basically a computational way to sift through the data and see which factors were the strongest predictors. And they found that absolute neutrophil count, the frequency of those LOX1 plus PD-L1 plus neutrophils, and PD-L1 expression were all highly associated with later disease severity.
Really? More so than other factors?
Well, get this. In their data set, these specific neutrophil features were actually better predictors than age or BMI.
Wow, that's surprising given how much we focused on age and BMI.
It is. Now, this is one data set, of course, but it strongly suggests that specific changes in neutrophil features, especially involving PD-L1 and LOX1, are present early and correlate strongly with who's likely to get severely ill.
All right, so these distinctive marker-positive neutrophils are found in severe cases, and markers linked to them appear early in those who eventually get sicker. This leads to the really critical question, the one this paper really tries to answer. Is SARS-CoV-2 itself directly causing these neutrophils to change? Or is this just a secondary effect of all the inflammation and damage happening during severe illness?
Right, the cause and effect question.
Right.
This is where they did some really elegant experiments in the lab. They took healthy neutrophils from healthy donors, the normal kind, normal density neutrophils or NDNs,
Yeah.
and they exposed them directly to SARS-CoV-2 in a dish. What they discovered was quite remarkable, actually. Simply incubating these healthy neutrophils with SARS-CoV-2 rapidly triggered LOX1 expression on their surface.
Rapidly. How
Fast. We're talking within just an hour.
Within an hour. Wow, that is fast. So does the virus have to infect the neutrophil and start replicating to cause this?
No, and this is a crucial finding. It really shifts how we might think about this interaction. The LOX1 induction happened even when they used inactivated SARS-CoV-2 particles, dead virus, essentially.
Just the particle itself is enough.
Exactly. This clearly shows that the virus doesn't need to replicate inside the neutrophil. The mere presence of the viral particle seems enough to initiate this change.
That's fascinating. Was it specific to SARS-CoV-2?
Good question. They checked. This rapid LOX1 induction occurred with multiple SARS-CoV-2 variants they tested. But importantly, it did not happen when they incubated neutrophils with influenza A virus.
Ah, so it's not just any virus.
Seems not. And this actually lines up with other observations that patients hospitalized with flu didn't show this specific elevation of PMN MDSCs, according to prior work they mentioned.
Okay, so how does the virus particle do this then? What's the mechanism?
So they dug into the how. They found that SARS-CoV-2 exposure led to neutrophil degranulation.
Degranulation again. Releasing their contents.
Exactly. Imagine a neutrophil as like a little bag filled with different tools in tiny internal compartments called granules. Degranulation is when it releases the contents of these compartments. They saw evidence that specific granules were releasing their contents, bringing molecules like LOX1 and PD-L1, which are normally stored inside, up to the cell surface.
So LOX1 and PD-L1 are inside these granules.
Apparently so, yes. And their experiments using a degranulation inhibitor showed that getting PD-L1 to the cell surface in response to the virus actually depends on this degranulation process.
So blocking the release blocks the PD-L1 appearance.
That's what they found. So the takeaway from these lab studies is pretty clear. SARS-CoV-2 can directly trigger healthy neutrophils to release specific contents, quickly bringing LOX1 and PD-L1 to their surface. And it does this just by being present, without needing to replicate inside the neutrophil.
Okay, this is a key step. The virus particle itself directly flips a switch in the neutrophil, makes it express these markers like LOX1 and PDL1. Now, what do these altered neutrophils actually do? What's their effect on the immune system's ability to fight back?
Right, the functional consequence. This is where they tested the function of these virus-exposed neutrophils. They put these neutrophils into co-cultures with healthy T cells, the key fighters the body needs to clear a viral infection.
Okay, see what they do to the T cells.
Exactly. And what they observed was significant. The neutrophils that had been stimulated by SARS-CoV-2 inhibited the proliferation, the growth, and multiplication of both CD4 and CD8 T cells.
Inhibited them.
Yes, and in a dose-dependent manner. The more of these altered neutrophils were present, the more the T cells were suppressed.
So they actively shut down the T cells. Was that suppression linked to those specific markers we discussed, LOX1 and PDL1?
Absolutely. They found a direct correlation. The more LOX1, ROS, and PDL1 the stimulated neutrophils had on their surface, the greater their ability to suppress T cell proliferation was.
So the markers correlate with the suppression.
They do. And the study provides strong evidence that the PDL1 expressed on these altered neutrophils is a key mechanism behind this suppression. They showed that this PDL1 was actually inducing death in T cells that weren't actively dividing.
Healing T cell.
Inducing cell death, yes. This could be a significant contributor to the T cell depletion, that lymphopenia seen in severe COVID-19.
Makes sense.
And importantly, they performed experiments using inhibitors to confirm this mechanism. When they blocked degranulation with that chemical inhibitor, Nexinhub-20, the neutrophils didn't become suppressive towards T cells.
Right, because the PDL1 didn't get to the surface.
Exactly. And even more compelling, when they used inhibitors that block the PD-1, PDL1 pathway, things like atezolizumab, similar to drugs used in cancer immunotherapy.
Yeah, those checkpoint inhibitors.
Right. Using those, they were able to rescue T cell proliferation and restore their ability to produce important signaling molecules like IFN gamma.
Wow. So blocking PDL1 stopped the suppression.
It did. This provides really compelling evidence, I think. SARS-CoV-2 directly creates these functionally suppressive neutrophils through degranulation, getting LOX1 and PDL1 to the surface. And that suppression largely happens because of PDL1 shutting down T cells.
Okay, let's pull all these threads together then. This deep dive reveals a pretty compelling potential pathway. The SARS-CoV-2 particle itself directly triggers neutrophils to degranulate and become these specific suppressive PMN MDSCs. These altered neutrophils then seem to actively dampen the crucial T cell response needed to clear the virus. Is that summing it up?
I think that's a great summary. Yeah, this research significantly advances our understanding. It moves beyond simply observing high neutrophil counts in severe COVID, which we knew. It reveals a specific virus-initiated mechanism involving degranulation and PDL1 expression on these cells that could directly contribute to why the body struggles to clear the virus and why some people get much sicker by impairing the antiviral T cell response.
And understanding this direct interaction, well, that could potentially point towards new therapeutic ideas, couldn't it?
It could, potentially. The findings suggest that perhaps targeting the formation or function of these specific PMN MDSCs, or maybe interfering with the PD-1, PDL1 pathway specifically on these neutrophils, that could be a therapeutic strategy worth exploring in severe COVID-19.
Interesting angle.
Now, as the source authors themselves point out, there are always limitations, right?
Of course.
While this study shows a very strong association between these LOX1 plus PDL1 plus neutrophils and severe disease and their early appearance, it doesn't definitively prove they are the sole cause of severity. Correlation isn't causation.
Right, the classic caveat.
Though the biological mechanism they've uncovered is highly plausible. Also, the precise role of LOX1 signaling itself in mediating the suppression isn't fully pinned down by this study. Is it just a marker, or is it actively contributing? That's still a bit open.
Okay.
And of course, experiments in a lab dish, well, they can't perfectly replicate the full complexity of what happens inside a living, breathing person.
True. Always a difference between in vitro and in vivo. But even with those caveats, this paper offers a powerful piece of the puzzle, showing a mechanism by which the virus might directly manipulate a fundamental part of our immune defense. And that could help explain the drastic difference in outcomes we saw for some people. So, what does all of this mean for you, listening?
It means that understanding these subtle but really significant ways a virus can interact with our basic immune cells, like neutrophils, that's absolutely critical to understanding the complexities of disease severity and potentially finding new ways to intervene.
Exactly. This research highlights that SARS-CoV-2 isn't just passively replicating and causing damage. It appears to be actively reshaping the immune response from the ground up by directly altering neutrophil function very early in the infection.
Actively reshaping it.
Yeah. And this raises an important question, I think, for future research and potential therapies, given that MBSCs can sometimes have beneficial roles like controlling excessive inflammation.
Why, they're not always bad. Not always. So is the goal to completely prevent their formation in COVID-19? Or is it more about selectively modulating their specific suppressive functions, like the PD-L1 pathway? And that might depend on how severe the infection is and what stage it's in. It's a nuanced question.
A truly fascinating look at how the virus seemingly manipulates our own defenses against us. Thanks so much for sending us this source and for letting us dive deep into it. Keep those questions and sources coming. We'll see you on the next Deep Dive. Thanks for listening today. 4 recurring narratives underlie every episode: boundary dissolution, adaptive complexity, embodied knowledge, and quantum-like uncertainty. These aren't just philosophical musings, but frameworks for understanding our modern world. We hope you continue exploring our other podcasts, responding to the content, and checking out our related articles at helioxpodcast.substack.com.